Movement of molecules between cells. So we've talked a bit about how molecules can move in and out of the cell by way of channels or just sort of floating, diffusing across the plasma membrane. Small molecules can do this quite well. Big molecules, most of what we're dealing with, can't do that. They're just too big. So you need to create some sort of a transport vessel to bring big things into the cell and to get big things out of the cell as well. Each one of these bubbles that you're going to use to bring things in or get things out is called a vesicle. This is a thin double layer plasma membrane. It's a lipid bilayer. It's otherwise fairly naked. It's just sort of your lipid bilayer for the most part. Note that it's made up of a lipid bilayer just like the plasma membrane. Remember too that like things can basically merge with other like things fairly easily and blend in with them. The process of bringing things into the cell is called endocytosis. Endo refers to in, cytosis is cells doing stuff. There's two forms of endocytosis that we should care about: phagocytosis and pinocytosis. Phago refers to eating, and pino refers to drinking. So phagocytosis is a process of cell eating things. Pinocytosis is the process of the cell basically drinking things. Pinocytosis brings in water. Phagocytosis brings in stuff. Step by step, let's go through how this works. You have some object. Forget about the arrows for a minute because I'm going to use the same drawing for in and out. You have something on the outside of the cell that the cell wants to bring in, or that the cell will be compelled to bring in, as in the case with viruses. This object makes contact with the outside of the cell. Once the cell makes contact with one of those protein molecules that looks for stuff and decides whether or not the cell wants to bring it in, if the cell recognizes it as something worth bringing in, what it'll do is it'll deform its membrane. It'll create a big dent around where those molecules are. This denting in is referred to as invagination. The membrane invaginates to create a dent for the object to rest into. From there, the dent gets bigger and bigger until the membrane can just pinch off and a bubble can be formed. That bubble, that double layer plasma membrane bubble that's created, that's now holding all of that stuff, is called a vesicle. Whatever cell receptors were here in the first place will remain intact over here. After that point, this vesicle is going to go forth and fuse with something, usually a lysosome. A lysosome is an organelle that has a bunch of digestive enzymes in it. It's good at breaking things down. So typically, when something's brought into the interior of the cell, it's big, fat, complex. Maybe it's something you want, maybe it's something you don't actually want. You were tricked to bringing in. Once it gets in the cell, you need to break it down into smaller, more usable parts, either for future metabolism or to just get rid of it. Either way, it fuses with the lysosome typically. Once the interior has been digested, the plasma membrane components will get recycled back up and fuse back into the original cell membrane. It probably doesn't fuse back into the exact same spot, but consider that whatever receptors it had studded on its surface that it brought in the first place is probably fairly evenly distributed across the cell membrane anyway. It's not terribly important. So, just again, a recap. Stuff binds to the outside of the cell. Cell says, hey, I want to bring that in. Cell makes a big dent around where that stuff is. That dent gets deeper and deeper, and the surface of it gets closer and closer until it pinches off, and this lipid bilayer, the plasma membrane, refuses with itself to stay smooth. That bubble of plasma membrane with the stuff in it, the vesicle, travels into the cell, makes contact at some point with lysosomes. Lysosomes have enzymes. They inject it into the vesicle, digest, break things down, make things usable. And then the plasma membrane can break back up, release those things into the cell, and come back to repatch up the cell membrane, to restore the original surface area of the cell membrane. For a short time, then, the cell has slightly less surface area to work with immediately after phagocytosis. And that surface area is restored when the vesicle that's now empty comes back and replaces that chunk. Pinocytosis is the exact same sequence of steps as phagocytosis. The only difference, really, meaningfully, practically for our purposes, is that you do the exact same process, but you don't have stuff. The cell simply decides it needs more liquid on its inside, and so it creates its invagination towards the aqueous exterior of the cell and brings in that liquid. The opposite of that process is exocytosis, moving things out of the cell. This is usually stimulated by some sort of an external force. The cell receives a signal that tells the cell, hey, you need to release this hormone out into the body needs it. Or, hey, we need this neurotransmitter, can you release some neurotransmitter for me? Or, hey, we need some more mucus on this membrane, we're starting to dry out, can you secrete more mucus? This also is used to export waste from the cell. So all life in the process of consumption and digestion is also going to end up creating some kind of waste. Some of it's going to be gaseous, some of it's going to be solid in the form of molecules, of non-gaseous molecules. You got to get that out. If you let it accumulate, it's toxic. Nobody wants to keep their waste on the inside. You got to get it out. So it's going to do the same process to get all that waste out. It's the opposite process of endocytosis. If you know and understand the steps of endocytosis, you write those in the opposite direction, and now you have exocytosis. The stuff of interest gets bound up by a lipid bilayer, just like the one you brought in when you were eating something. The outbound molecules are collected into that lipid bilayer vesicle, and they'll migrate towards the surface of the plasma membrane, or towards the interior of the plasma membrane. Because this stuff is made up of the same stuff as this, they're able to merge. There's also extra proteins called fusion pores that are bound to the cell membrane that really help anchor on the vesicle once it arrives. When the vesicle meets up with that fusion pore, it's able to merge with the plasma membrane. As it merges with the plasma membrane, the edges push outward, that bubble turns into a dent and releases that stuff into the outside of the cell. Okay? There's a video that I've linked to that shows you the process of the different kinds of diffusion as well as vesicular transport. That's listed both in this item on Blackboard underneath here with the slides in this video. Also, it's going to be listed in a separate item on Blackboard towards the bottom of the page, along with a list of all the other videos that I'll be referencing in this module.